It might seem crazy what I'm about to say. It's about prokaryotic replication of DNA.
First the double helix unwinds by initiator proteins and helicase. At the origin of replication with a fork that points the way. DNA replication.
SSC protein stabilize these strands into isomerase, releases tension.
DNA replication.
These two strands are template strands that are needed for new strand conception. DNA replication.
So two strands are anti-parallel but are replicated in the 5 to 3 direction.
DNA replication.
There's a leading and lagging strand used for nucleotide collection. This is semi-conservative, which means that the parental and new strands form a pack. The parental strand is where the new strand finds. This whole replication process doesn't take much time. Here's why. DNA replication.
Both strands start with short RNA primer that DNA primase synthesized.
DNA replication.
Then DNA polymerase moves from factor 3 to add nucleotides. DNA replication.
One strand is the lagging strand made opposite to the fork direction.
DNA replication.
It has to wait for the fork to move, so it's made in a discontinuous succession.
Lagging strand replicates continuously, has to wait for more DNA to be unwound because it has to move from 5 to 3 so the new DNA strand can be bound. This strand is replicated in sections. Okazaki fragments are what these are called because a template strand also has to read a 5 to 3 direction. This is how it's solved. I said DNA replication, the leading strand and replication form move in the same direction.
DNA replication.
Template strand reads 3 to 5, so there's no need for segmentation. DNA replication.
The leading strand is replicated continuously.
DNA replication.
The leading strand uses DNA primase, RNA primer, and polymerase 3.
DNA replication. Okazaki fragments are joined to make one continuous strand.
DNA replication.
In order to do this, polymerase 1 lends a hand. DNA replication.
It removes the RNA primer and replaces it with DNA.
DNA replication.
Resulting in a new strand that is here to stay.
Single-stranded gaps still exist between the fragments even after the primers have been replaced. Whole DNA strands are needed for completed replication, so these gaps are closed by the DNA ligase. This process keeps repeating as the DNA continues to unwind.
DNA replication.
The semi-discontinuous model is the name that is assigned. DNA replication.
Because the leading strand is synthesized continuously.
DNA replication.
While the lagging strand is synthesized discontinuously. DNA replication.
Sometimes in the replication process there is a mistake.
DNA replication.
But fixing them can be as easy as a piece of cake. DNA replication.
Polymerase 1 can fix them with 5 to 3 or 3 to 5 exonucleus activity. Polymerase 3 fixes them with 3 to 5 exonucleus activity.